This recording is about the physiological formation of sweat for sweat glands, sweating, and the changes in sweating due to the CFTR, the cystic fibrosis chloride channel. I have a note here on what you need to understand in order to follow this video. We're talking about sweat secretion from eccrine sweat glands. They secrete water with dilute sodium and chloride. Their function is for cooling. We are not talking about apocrine sweat glands, which secrete skin oils and other substances into hair follicles. The major sources are these two articles, but I also took material from some other sources. This shows an eccrine sweat gland in an image from Anatomy and Physiology, open stacks. Each sweat gland is coiled and then has a portion that releases sweat out onto the skin surface. The sweat gland has two parts. The initial part produces the salty fluid, and this later part reabsorbs the salt so that salt is not wasted and lost. There are two regions of a sweat gland. The two regions are the secretory region, where chloride, sodium, and water are secreted, and then the reabsorptive region that recovers sodium and chloride. The steps are that first chloride is moved in. Second, sodium follows in between cells in a paracellular pathway, attracted by the negative charge of the chloride. And third, aquaporins in the membrane allow water through, producing a salty fluid that is isotonic. It is the same concentration as the extracellular fluid up here and inside. In the reabsorptive region, the chloride and the sodium are reabsorbed, but the water is left behind. So the result is a sweat that is a dilute fluid with water, but it has low sodium and chloride. You can't get back all of the salt that you produce. There are two endocrine signals that cause sweating. There's thermal sweating, and we won't talk about it, but that's driven by acetylcholine from the nervous system. And then there's stress and excitement sweating, which is driven by noradrenaline from the adrenal gland. Let's first talk about the secretory region, which produces the salty fluid. The first step is the sodium-potassium pump, which uses an ATP to move sodium out, and of course also potassium in. Now the potassium is continually recycled. The sodium drives the NKCC transporter, that's third, moving one sodium, one potassium, and two chlorides into the cell. Remember, this is the extracellular fluid here. Down here is the lumen. That chloride then can move through a non-CFTR chloride channel. That's 5, which brings chloride into the lumen. That chloride will attract any sodium nearby. And in this region, in the secretory region of the gland, sodium can move through the tight junctions. So these are tight junctions, and tight junctions can be tightly sealed. The different proteins allow some tight junctions to be tightly sealed so that no water or ions can pass through them, but some are selectively permeable just to sodium. And so these allow sodium, remember the sodium is high outside because of the sodium-potassium pump, so that allows this sodium to move in where each sodium winds up being attracted to a chloride. That attraction to sodium and chloride means that sodium, even though it could go that way, it almost never does, because each sodium coming in winds up being attracted to all this negative charge. And so we have a net movement of sodium in. So that's number 6. And then 7, because of that sodium, water easily leaves the cell to form hydration shells around the sodium and the chloride. Because each water that moves in winds up being part of a hydration shell, even though the water could go in either direction through this aquaporin, a water channel, you get net movement of water into the lumen. Because water is leaving the cell on this apical side, the basal side, so basal side and apical side. Basal side is always the side near the basement membrane and the extracellular fluid. The apical side is the side near the lumen. So 8, we also have water coming in, and that produces an isotonic solution of sodium, chloride, and water. Same concentration of sodium and chloride as on the extracellular fluid side, so same concentration, isotonic to extracellular fluid. But the effect of all of this is to continually move chloride, 1, 2 water, and 2 sodium, and 3 water into the cell, into the lumen. Now, a problem with that is that sodium and chloride is normally valuable. In humans, over most of our evolutionary history, have not had access to enough sodium. We get plenty of potassium chloride in our food, but sodium can be in short supply, since plants don't tend to have a lot of sodium. So in the absorptive region, we recover sodium and chloride, but not water. The steps are, again, the sodium-potassium pump creates a high sodium concentration outside, low inside. Again, the potassium here is recycled continually. In the absorptive region, then there is a sodium channel and other sodium transporters that allow sodium to move across. And there's also a cystic fibrosis channel. And so we have one, the low sodium inside brings sodium into the cell, so we remove sodium. And with that movement of sodium away, there are chlorides that are not attracted to any sodiums because the sodiums are gone, and so the chloride follows that charge out through a chloride channel. And this chloride channel is the cystic fibrosis chloride channel, the CFTR. It is regulated. If you have excess salt, you can produce saltier sweat, but if you're conserving your salt, and normally humans are conserving at least some salt, you do this. So this is regulated, and that's an important part of the CFTR channel function. The CFTR is regulated by cyclic AMP. So it's opened by cyclic AMP. In the absence of cyclic AMP, you have some of this happening, potentially, although I think that one may be closed in the absence of cyclic AMP2, but I haven't found it. In any case, without cyclic AMP here, this channel is closed. And so it's regulated. There's a G protein coupled receptor system that causes adenylate cyclase here to make cyclic AMP. And so when your salt concentrations are high, you inhibit this pathway, no cyclic AMP, you don't recover the chloride, and you can produce saltier sweat. But when you need to recover it, this pathway is activated. So it turns out that cystic fibrosis patients produce sweat that has abnormally high chloride. The reason is that they have a non-functional cystic fibrosis chloride channel. There's a mutation that makes that be absent. And so even though you can still have this part, sodium out, sodium following, with no chloride following, very quickly the sodiums that might leave instead are attracted to the chlorides that you can't get out of the lumen. And so measuring chloride in the sweat, which also holds on to a lot of the sodium, means that you can't reabsorb it, and so without the reabsorption of sodium and chloride, a cystic fibrosis patient produces water and salty NaCl, high concentrations. Now, there's a potential adaptive benefit of cystic fibrosis during cholera epidemics. And that's because the colon has a different role for the CFTR channel, the cystic fibrosis chloride channel. Normally in the colon, you need to reabsorb all that salty fluid that kept your gut contents fluid. And so you have a series of ways for sodium to get from the lumen into the cell. There are sodium channels. There's a sodium and chloride co-transporter that's driven by the low sodium concentration in the cell from the sodium-potassium pump. There's a sodium proton exchanger that moves acid in, and there's also a chloride bicarbonate exchanger that brings chloride back and other avenues. So normally you're reabsorbing sodium and chloride and moving them into the extracellular fluid in the colon. But during cholera, that gets messed up. The cholera toxin moves into a cell. It's endocytosed into a cell by binding to a surface protein. It goes through several steps, and the key one is that it activates a G protein subunit to keep it bound, to keep that G protein subunit bound to adenylate cyclase. And so adenylate cyclase keeps making cyclic AMP. You can have 100 times normal cyclic AMP when there's cholera toxin. That opens up this cystic fibrosis channel. So that cyclic AMP binds again to the cystic fibrosis channel. It also opens a potassium channel. And so both of these are activated. The net effect, remember there's normally high potassium inside the cell, the net effect is that potassium leaves quickly and chloride leaves quickly, activated in both cases by this cyclic AMP. And a cystic fibrosis patient has a mutation that has a non-functional chloride channel. And without the chloride channel, even though some potassium will leave, it won't keep going because chloride winds up trapped here. There's not enough chloride in the lumen to attract this potassium. And so a heterozygote for cystic fibrosis loses less chloride and is protected from the diarrhea of cholera toxin. So you might wonder, why have this system at all? Because it seems like all it does is make you susceptible to cholera. Well, this whole system is normally activated when potassium and chloride are in excess. So if you've had excess chloride and excess potassium, that activates this system to allow you to get rid of some of that potassium and chloride in feces. And it probably also does things like keep the feces moist enough. So there may be other functions there. So that's a hypothetical potential adaptive benefit. The cystic fibrosis gene is more abundant, that mutation is more abundant than you would expect if it's a pure negative. So there may be a heterozygote advantage, less diarrhea. with the huge amount of chloride and potassium that gets out during cholera creates terrible diarrhea, and people die of loss of solutes, loss of electrolytes. So if you prevent that loss of chloride and potassium, you protect against that diarrhea, and there's a survival benefit.